Hello, advanced biology students, and on to our next part of our DNA unit lecture outline. Today we're going to be talking about transcription and translation, both of which are very important processes that tap into the coded instructions that are found inside the DNA molecule. Now, you're very used to hearing that DNA is the blueprint for life. It carries all the instructions for building you, building other living things. Transcription and translation are the processes through which we take the information that's inside of DNA and build something with it. So we're going to be talking about what those processes are and how they work. First key point, we're first going to focus on transcription. And the first major point you need to think about is DNA is a very, very large molecule, and we find it where in the cell? That's right, in the nucleus. And the nucleus is essentially a container that holds it, and DNA is too large to get out. So we have to make short copies of the information that's found on DNA when it's needed. It's kind of like having a textbook for a class. You only read a chapter at a time. We have to make short copies of chapters of DNA when we need them, because you don't need all the chapters at one time in the cell. And how do you photocopy those short segments of DNA? A process called transcription allows for us to make a short RNA copy of it. Okay, so that's what we're going to be focusing on first, is how RNA carries DNA's instructions. So how do we figure this out? Watson and Crick define the central dogma of molecular biology, what we call the central dogma of biology, which is that DNA goes to RNA, which goes to protein. And I like to add on that protein goes to phenotype. All of your phenotypes are in some way dependent on a protein involved in the process. First of all, replication, which we covered in our last video segment, copies DNA. This happens during synthesis of the cell cycle. You need that for when you make new cells. Transcription, however, converts DNA message into what we call an intermediate molecule called RNA. Intermediate in that it has a short-lived purpose to go from the nucleus out into the rest of the cell, into the cytoplasm where proteins are being made. And then finally, translation. Think of what a translator does. They translate from one language to another. Translation interprets an RNA message and then makes a protein from that. So it translates it from the language of nucleotides, which we learned were the building blocks of DNA. And they are also the building blocks of RNA as well. But translation takes those nucleotides and converts it to a new language, the language of amino acids that we string together to form proteins. So in prokaryotic cells, these processes take place in the cytoplasm. But in eukaryotic cells, we know that eukaryotic cells have a nucleus, so the processes are separated. Replication and transcription both take place in the nucleus in eukaryotic cells, and translation occurs in the cytoplasm. So RNA, its major purpose is to act as a messenger between the nucleus and protein synthesis that's taking place in the cytoplasm. And you can see in the picture below, you have these short segments of what we call messenger RNA. We'll get more into what messenger RNA is in just a moment. But RNA is also a nucleic acid, where DNA stood for deoxyribonucleic acid. RNA stands for ribonucleic acid. They're very similar molecules, but they do have some differences. RNA is different from DNA in 3 significant ways. First of all, the sugar in RNA is called ribose, not deoxyribose. And if you'll notice below in the picture on the right, this deoxyribose molecule, you'll see the hydrogen that's highlighted in red. If you look at the ribose on the right, the similar highlighted region, what's the difference between these two sugars? If you'll notice, ribose has an additional oxygen. That's actually where the name deoxyribose comes from. It's that it's deficient in oxygen, so it's short 1 oxygen when you compare it to ribose. Okay. So first of all, the sugar in RNA is called ribose and not deoxyribose. The second major distinction between RNA and DNA is that RNA has the base uracil in place of thymine. So instead of A, T, G, and C, RNA has A, U, G, and C. Very similar molecules, but the nitrogenous base uracil is found instead of thymine. And then finally, RNA is single-stranded, not double. Transcription makes 3 kinds of RNA. Transcription copies sequences of DNA in the nucleus, pictured here below. See the nucleus right here. DNA has to separate. Those hydrogen bonds that hold those nitrogenous bases together have to open up, and then short segments of RNA are transcribed or copied from that. And transcription copy sequence of DNA are 1 gene long. and are catalyzed by RNA polymerase. Whereas DNA polymerase govern the process of replication, RNA polymerase governs the process of transcription, where RNA polymer is made. DNA first has to unwind at a specific site, a gene in particular. And using one strand of DNA, so only one strand of DNA gets used in this process. And this is different from replication in that both sides of DNA are copied during replication. But in this case, only one strand of DNA is used. What we say complementary strand of RNA is produced, meaning that it matches with that DNA strand that it's copying. So the RNA strand will detach and DNA will reconnect. So notice in this animation below we have DNA. It's held together by those hydrogen bonds. But you'll notice right below it, there's RNA polymerase. This is the enzyme that helps start this process. And you'll notice in the far left, we have this region called a promoter. This is kind of the short sequence of DNA that signals to RNA polymerase that it needs to attach here. And that's the start of our gene. And as DNA gets separated, RNA polymerase, and you also have helicases that help open up the helix, starts building together nucleotides that match the DNA strand of interest. But in this case, you'll notice there's a uracil that pairs up with an adenine instead of a thymine. So I remembered that nitrogenous base is different. And it continues to do this along this whole strand until it reaches a region called the terminator. But that signals the end of a gene and signals to RNA polymerase that it needs to detach, and you end up with a separate segment of messenger RNA, as seen below. So transcription produces three kinds of RNA. The first kind is one we'll spend a lot of time talking about, and that's messenger RNA. We use mRNA as a shorthand way of abbreviating that. This is the code that's used for translation to make proteins. So this is what determines genes and traits and things like that. Ribosomal RNA is also formed from transcription. This forms parts of the actual ribosome. And then finally, transfer RNA, or tRNA, is an important type of RNA that helps bring amino acids from the cytoplasm to a ribosome to help make a growing protein. So all three of these kinds of RNA are really important in translation, which is the next process that we'll talk about in just a moment. So transcription is really similar to replication in a couple ways. First of all, they both occur in the nucleus. Both involve the unwinding of DNA and the breaking of hydrogen bonds. And both involve complementary base pairing, meaning the nitrogen-containing bases have to match up in order for this process to take place. Now, on to translation. How do we take this code and build something useful with it? So amino acids are coded for by messenger RNA base sequences, and translation converts mRNA into polypeptides or proteins. So you need to know the term codon and anticodon. We're first going to focus on what a codon is. Basically, we don't read messenger RNA or DNA or any of these as individual bases. The way it gets read are in what we call triplets, which are 3 nucleotides long. Like you'd see right here, this is A, U, and G. And then AAC. Think of it as words that are being constructed in DNA. So only three bases at a time get read. This is what we call a codon. And each codon is the code for an amino acid. So for a building block for a protein. Now RNA can code for 64 different combinations because there are 3 different bases in a codon, and then you have 4 different potential bases that it could be. So you get these 64 different combinations. The chart below actually shows you all these different combinations, which is plenty to cover the 20 amino acids that we need to build proteins in the human body and most other organisms. Code. Many amino acids are coded for by more than one codon, and we also have these special codons called start codons and stop codons. These signal the start of translation, where should the ribosome actually attach, and then also stop codons. It signals the end of the amino acid chain and tells the ribosome to release that. And this code is universal, and it's the same in almost all organisms. And it suggests a common ancestor. This is actually one of our evidences for evolution, in that we all have the same DNA code, and it codes for the same amino acids for the most part. And it means also that scientists can insert the gene of one organism into another to make a functional protein. This is actually the basis of biotechnology and how we use living things as a tool or technology, the basis for cloning and recombinant DNA. All things are going to be talking about soon as well. So amino acids are linked together to become a protein. So you have two important tools you need to translate a codon into an amino acid. So you need the ribosome, which is the site of protein synthesis. You can see below that a ribosome has a large subunit and a small subunit. And you'll notice there are these three little binding pockets. So the large subunit binds to tRNA, which you may not remember just yet, but you'll get used to this. tRNA actually holds the amino acid. It actually holds the protein building block. And then you also need the small subunit, which is where messenger RNA goes to. Now, if you remember, messenger RNA gets made in transcription in the nucleus and gets sent out, because it's a shorter copy, gets sent out to the cytoplasm where it then binds to the ribosome. You also need the tRNA, which is a very important tool in this process too. This carries free-floating amino acids from the cytoplasm to the ribosome. Now, where do you get these amino acids, you might ask? Remember, you eat food that contains proteins? Well, during digestion, we break these down into the building blocks, which are amino acids that make their way into the cells where they can be recycled to make new proteins. Now, the tRNA attaches to a specific amino acid and has a 3-letter anticodon that recognizes a specific codon. So those codons you learned about in messenger RNA match an anticodon, and it's very similar to DNA in that if you have an adenine on the messenger RNA, it has to match up with not a thymine, because remember this is RNA, it has to match up with a uracil. So A and U would base pair together. G and C still base pair the same way. The only major distinction here is that uracil. But there's an anticodon on tRNA that recognizes the codons on messenger RNA. And they actually kind of meet in the ribosome itself and briefly base pair. So translation occurs in the cytoplasm of the cell, and the messenger RNA first has to bind to the ribosome. And the ribosome pulls the messenger RNA strand through one codon at a time. As you can see, it kind of gets fed through like a ticker tape. And as the messenger RNA goes through the ribosome right here, tRNAs meet and base pair. And as this is happening, you'll notice, here's our anticodon and codon. The exposed codons along the way attract complementary tRNAs bearing an amino acid. And they bring them together so that as they go through the ribosome, the amino acids are close enough that they can bond together. And see how these two amino acids are now linked together? And the tRNA molecule that now has delivered its amino acid leaves to find another amino acid out in the cytoplasm. And the ribosome continues to do this down the entire messenger RNA sequence, attaching more amino acids until it reaches a stop codon, which you see right here as UGA. And notice we have this growing polypeptide chain that's linking together and leaving the ribosome. Now, I know this might be a challenge to understand. Don't worry, tomorrow we'll be doing a simulation of this, and you'll be practicing building your own protein chains. Please come in with questions. Don't forget to write the questions down in your lecture outline for tomorrow.